I'm Haleigh, and I'm a junior at Northwest University. And this summer we did research at the Allen Institute through UW's and SSSP program to verify viral vectors.
This summer we had the privilege of working at the Allen Institute within the cell types project led by our principal investigator, Dr. Lein, and we're on a part of a team led by Dr. Ting, where we were mentored by scientist Peter Chong throughout the 8 weeks.
The Allen Institute was designed to help answer some of medicine's most pressing questions, and specifically, the Allen Institute for Brain Science was designed to create a better baseline for how the brain functions under healthy conditions so that progress can be made to work to see what goes wrong in diseased brains and how to cure such neural states. Our project objective was to verify various viral vectors that a collaborator, Jimena Pizarraya, made in fluorescently tagging medium spiny D2 or D1 neurons in a striatum. This is important because D1 and D2 MSNs are involved in various different neurodegenerative diseases when they become dysfunctional.
The basal ganglia has been associated with many different diseases and conditions, from anywhere from addiction, Parkinson's disease, to obsessive compulsive disorder. And if we were able to target D1 and D2 medium spiny neurons specifically, then we can move forward to better understanding these neurons through methods like patch clamping and cellular reconstructions. And this could also be a huge step into clinical applications like gene therapy.
In order to verify these viral vectors, this is the basic method that we followed, which began with retroorbital injections. To perform a retroorbital injection, the animal is first anesthetized, then the operator pulls back on the skin surrounding the eye so that it appears to be bulging, though being careful not to obstruct the trachea or the cervical vessels. Then the operator injects the needle into the medial campus and pushes forward until it reaches the retroorbital sinus where the virus is then injected.
We then waited about 3 weeks in order for the virus to target the area of interest and infect the cells, where we then performed a terminal surgery called a perfusion. And a perfusion is a procedure which replaces all existing blood in the brain and replaces it with a solution called NMDG, which helps to keep the neurons alive and firing even after dissection. And to do this, we first anesthetized the mouse, where we then opened up the abdominal cavity, then snipping the right atrium of the heart, where we then injected the NMD solution into the left ventricle until the NMDG solution was perfused throughout the entire body. We then dissected the brain and prepared it for either IHC or electrophysiology.
In order to verify the viruses, we used immunohistochemistry. So IHC uses antibodies and fluorescence to tag to and light up your target antigens or proteins. So after we dissected out the brain, we fixed it in paraformaldehyde. Then we decided to cut the slices sagittally because we knew that D1 neurons projected from striatum to substantia nigra and D2 from striatum to globus pallidus. So sagittal slices revealed all 3 areas in one slice. Then we picked out our favorite slices and we applied a few different antibodies to them. DART32 was an antibody that bound to all D1 and D2 medium spiny neurons so that we had something to compare the viral signal to. Then we applied anti-GFP, which enhanced the viral signal. After applying the secondary antibodies, we looked at the tissue under fluorescence and were able to compare and see any co-labeling that occurred.
We could also look at the electrophysiological properties of these neurons by analyzing its behavior and then identifying its neuronal type. And to do this, we performed a method called patch clamping, which involves taking a pipette with a microelectrode down to the cellular level, like we did here in that picture. And then we applied a suction to form a gigaohm seal of the cell membrane. And then we could actually apply current in order to stimulate an action potential. And from this propagation, we can then look at the behavior and identify a cell type. And so we know that D1 neurons are less excitable and D2 neurons have a greater firing frequency. And so from this baseline knowledge, we can then compare these behaviors to those of diseased neurons and compare the two.
We tested a total of 4 different viruses. 2 were successful in labeling all D1 and D2 medium spiny neurons, and 2 sparsely labeled them. This one, virus 2011, sparsely labeled the neurons. And we know that because when we were looking at the image under DART32, which labels all D1 and D2 MSNs, you see the striatum and globus pallidus brightly lit. Unfortunately, this slice was more lateral, so it doesn't have a substantia nigra, but that both D1 and D2 neurons go through the lobus pallidus, so we know that both are there. Comparing that image to that under anti-GFP, you don't see any extra fluorescence in the same two areas. The whole slice is the same color, so we know this was just background. And this is further confirmed when we look under the images under 20X. So all of those little white dots there under dark 32 are the somas of D1 and D2. But then with anti-GFP, you only see a couple of neurons being labeled. Sparsely labeling isn't necessarily a bad thing because other researchers who use techniques like electrophysiology and reconstructions need neurons to be sparsely labeled. They need to see the somas, the dendrites, and all the axons.
The 2025 virus yielded more successful results. Dark 32 is the antibody that stains MSNs within the striatum, and this can be seen with signaling projecting on the substantia nigra and the globus pallidus. The anti-GFP antibody was the viral enhancer, which also similarly you can see labeling within the striatum with projections to substantia nigra and globus pallidus. So the co-labeling image suggests that the 2025 virus targeted both D1 and D2 medium spiny neurons. And studying D1 and D2 medium spiny neurons is important for us when trying to understand Parkinson's disease, which is a degenerative neural disease that disrupts the normal neural circuitry, linking thoughts and actions, often causing tremors and difficulties in movements. And this affects around 10 million people in the world today, and over 60,000 Americans are diagnosed with this disease every single year. And the mechanisms behind the development of symptoms of PD are unknown. However, we do know that in patients who develop Parkinson's disease, symptoms start to develop when 50 to 60% of dopaminergic neurons experience cell death, and 70 to 80% of dopamine in the striatum is depleted.
Medium spiny D1 and D2 neurons are part of the basal ganglia circuit, which requires dopamine, helps facilitate your body's movement, learning, emotions, habit forming, etc. So when there's a depletion of dopamine, the medium spiny D1 and D2 neurons become dysfunctional, thus leading to an interruption in your brain's circuitry and eventually to the symptoms of Parkinson's. However, the mechanisms of exactly how this is occurring is still unknown. So our project in verifying these viral vectors is useful because now we can target these cell types to further understand them and how they function normally, to see how their dysfunction leads to things like Parkinson's, and now the possibility of gene therapy is available. We would like to acknowledge all the people who made this program possible, specifically Dr. and Mrs. Ellen Boken and Sylvia Sabachin and Julie Fold for spending so much time with us and making sure that our internship was as amazing as possible.
And also our PI, Dr. Lein, our team leader, Dr. Ting, and also our mentor, Peter Chong, who taught us valuable life and lab lessons.